Okay, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, right, so we're currently in the process of discussing inflammatory pain, and we've discussed that there are two major components to inflammatory pain. There is the tonic, prolonged firing of the polymodal nociceptors, which are attached to C-fibre, primary nociceptive afferents, okay? And then there is also the sensitization of all of the different types of nociceptors, so the thermal and mechanical nociceptors attached to the A-delta fibres, and also the polymodal nociceptors attached to the C-fibres. Okay, and we're currently looking at the mediators within the inflammatory milieu that mediate this, these two phenomena. Okay, right, so we're currently in the process of discussing prostaglandins, and the one that we're specifically looking at is prostaglandin E2, and we've seen how it's going to activate the classical GS cascade within nociceptors, basically, and we want to see how this is going to cause those two phenomena, and we're currently looking at the sensitization. Okay, so we've seen how you're going to get the activation of protein kinase A enzymes. These catalytic subunits here are now going to phosphorylate certain targets. One of those is the NaV1.8 alpha subunit of voltage-gated sodium channels, which, when phosphorylated, is going to change its threshold potential to make its threshold potential lower, basically, so that the receptor potential, the amount of receptor potentials that you need in order to actually excite the nociceptor into firing an action potential is going to be reduced, and that's going to underlie allodynia and hyperalgesia. Okay, one of the other things that is known about how prostaglandin E2 works is that these catalytic subunits of protein kinase A are actually going to phosphorylate TRPV1 subunits of TRPV1 channels. Okay, now what has actually been found is that the TRPV1 channel is associated in a complex with protein kinase A enzymes that are inactive. Okay, so let me just explain this. Okay, so firstly let me draw the TRPV1 channel here. So here is our TRPV1 channel, which is of course a tetramer of 4 TRPV1 receptor subunits. Okay, and basically it has been found that the TRPV1 channel here is going to be bound to a protein known as an ACAP. Okay, and specifically it's bound to ACAP79, which is also called ACAP150. Okay, so this is ACAP79, or you can also call this ACAP150. So what is an ACAP protein? Well, an ACAP protein stands for A kinase, referring to protein kinase A, anchoring protein. Okay, it's a protein which combines to certain forms of protein kinase A. Now, it can't just bind to any old form of protein kinase A. It specifically binds to protein kinase A known as type 2 protein kinase A. So what does that mean? Well, this refers to an inactive protein kinase A. So it refers to one of these tetramers up here that we've drawn, and it refers to protein kinase A tetramers where the regulatory subunits here are type 2 regulatory subunits. So basically, if you've got one of these protein kinase A tetramers that's constructed out of 2 type 2 regulatory subunits, like so, then that's known as a type 2 protein kinase A enzyme in the inactive form. And the key thing here is that these type 2 regulatory subunits are capable of binding to A kinase anchoring proteins, binding to ACAPs, whilst the type 1 regulatory subunits are not capable of doing that. And indeed, protein kinase A inactive tetramers, which are produced out of type 1 regulatory subunits, are known as type 1 protein kinase A enzymes. Okay, so here then, this is our type 2 protein kinase A enzyme that is bound to the ACAP79 or 150, whichever you prefer to call it, which is also bound to the TRPV1 ion channel here. Okay, so this is protein kinase A2 here. And the idea then is that when the cyclic AMP is produced, Again, the exact same thing is going to occur. Cyclic AMP will bind in these binding sites, causing a change in conformation in the regulatory subunits, and those will then release the catalytic subunits. And now the catalytic subunits are already extremely close to the TRPV1 channel. Okay, and now all they need to do is move over to the TRPV1 channel and now phosphorylate the TRPV1 proteins that make up that TRPV1 channel. And then what's that result in? Well, basically it results in the sensitization of the TRPV1 channel, so that it now opens at lower temperatures. So remember, previously TRPV1 required temperatures that were greater than 45 degrees Celsius for it to change conformation and open. Okay, once it has been phosphorylated by protein kinase A catalytic subunits, it will open at lower temperatures, and this therefore underlies allodynia. Okay, so this is another part of sensitization. This is specifically, of course, thermal allodynia in the thermal nociceptors and also the polymodal nociceptors. Okay, right. So there's another way that the prostaglandin E2 acting through this GS cascade can cause sensitization of nociceptors. Okay, right, what I now want to turn my attention to is looking at the mechanism by which the activation of this GS cascade is going to lead to the C-fiber attached nociceptors, those polymodal nociceptors, firing rhythmic action potentials, okay, similar to the way the heart fires rhythmic action potentials. And in fact, it uses the same ion channels as the heart uses to generate rhythmic action potentials. Okay, so let's talk about these very special ion channels. Okay, right, so the star here is the hyperpolarization activated and cyclic nucleotide gated ion channels. Okay, so hyperpolarization activated, it's a very long name, but it's abbreviated down to something very short. Hyperpolarization activated cyclic nucleotide gated cation channels. So cyclic nucleotide gated, which refers to the cyclic AMP that's going to have an effect on it. Cyclic nucleotide gated cation channels. Okay, and for short, the hyperpolarization activated and cyclic nucleotide gated channels are abbreviated down to the HCN channels. Okay, so let me discuss then the structure of an HCN channel. Okay, so HCN channels are once again tetramers. Okay, so if I draw the structure here, here is the HCN channel sitting in the plasma membrane like so, and again it's going to be constructed out of 4 separate subunits. Okay, and in fact the membrane spanning topology of the individual subunits is going to look exactly like that membrane spanning topology we saw for the TRP channels so long ago now. Okay, so this is what an HCN channel looks like, a hyperpolarization activated cyclic nucleotide gated cation channel. Okay, and if we pull out an individual HCN channel subunit here, let's say we'll have this one in purple here, okay, again, the membrane spanning topology looks like so. You have 6 membrane spanning alpha helices, 1, 2, 3, 4, 5, and then you'll have a p-loop between the 5th and the 6th, so here's the 6th, and then here's the amino terminus, and then here is the C terminus. Okay, right. Now when you want to build an HCN channel, there are multiple different types of HCN subunits that you have available in order to build a tetramer. And in fact there are 4 different types of HCN channel subunits that are available, okay, which are very nicely labelled up. HCN1, HCN2, HCN3, and HCN4. The type of HCN channel that is going to be extremely important in setting up the rhythmic action potentials firing along these polymodal nociceptors is going to be HCN2. Okay, so we're going to be talking about HCN2 homotetramers, where you have put 4 HCN2 subunits together to make a homotetramer, and that's your HCN2 channel. Okay, so these are the ones that are extremely important in the polymodal nociceptors. Okay, right. So now the key thing, that's the structure of the damn thing. Let's now look at the key thing, which is what does it do? What does it when does it open, and what does it allow to come through it? Okay, well, what it allows to come through it is the easy part. It allows sodium ions to come through it when it's open. So when it's in the open conformation, it allows a depolarizing current of sodium ions to come from the extracellular fluid into the cytoplasm. Okay. Now, what actually activates it to open is the more complicated part. It is activated to open by a very unusual thing, hyperpolarization. Usually, when we talk about voltage-gated ion channels, they are activated by depolarization, not hyperpolarization. Okay, remember back to the voltage-gated sodium channels. In order to activate the voltage-gated sodium channels, we needed the electrical potential difference across the cell membrane to be moved up to that threshold potential of negative 40 millivolts. That was a depolarization. Okay, these HCN channels are actually activated by you taking the electrical potential difference down, okay, to some hyperpolarized threshold potential, and I'm afraid I don't know what the normal number for that is, but some hyperpolarized threshold potential down here, basically. Okay, then what will happen is when they open, in response to this hyperpolarization, they'll allow sodium ions to come into the cell, and therefore they will take the electrical potential difference back up, basically, is the idea. Okay, now, basically, these are also interfered with by cyclic AMP. Okay, so not protein kinase A. This action of prostaglandin E2, this isn't dependent on protein kinase A. This is an effect of the cyclic AMP directly. So forget protein kinase A. It's cyclic AMP that's going to be important in affecting these things. Okay, so what can happen is cyclic AMP molecules combined to the HCN2 subunits within the HCN2 channel. Okay, and what they can do is they can change the electrical potential difference at which it will open. Okay, and they can move its threshold potential upwards. Okay, maybe they can move it right up to normal resting membrane potential. Okay, so we're now going to change the way this opens so that it's going to open at or nearby normal resting membrane potential. Okay, so we're going to depolarize the threshold potential for the HCN channel, basically. Okay, now what's the effect of this? Well, if we think about these now being in these free nerve endings of the nociceptor. Okay, so let's have our HCN2 channel here. Okay, now, whenever the electrical potential difference across the cell membrane of this free nerve ending is at resting membrane potential, negative 65 millivolts, these things will open and produce a receptor potential by allowing sodium into the cell. Okay, so whenever the electrical potential difference is normal, these things will then produce a receptor potential, basically, and that can then drive an action potential, basically. So all the time, these things are going to be tonically activating the polymodal nociceptors to fire action potentials, is the idea. Okay, and that's because they can't ever not stop doing this when the electrical potential difference across the cell membrane is at this resting value of negative 65 millivolts. Okay, so that having this channel now activated by resting membrane potential means that there now is no stable electrical potential difference across the cell membrane of this cell anymore, because as soon as you get down to what would have been the stable electrical potential difference across the cell membrane of negative 65 millivolts, this channel is now going to open and produce receptor potentials, which are then going to drive action potentials. So this is what leads then to this rhythmic firing of the polymodal nociceptors, because whenever they're not firing, this will now activate them to fire, basically, is the idea. Okay, so this might underlie how prostaglandins can make these C fibers fire just tonically, basically. Okay, right, so what we now want to discuss is the non-steroidal anti-inflammatory drugs and how they work. Okay, now we made a comment on this earlier when we were actually discussing the synthesis of prostaglandins. So the non-steroidal anti-inflammatory drugs. Now, as their name suggests, they have very powerful anti-inflammatory effects as well as their ability to relieve pain. Okay, and that's because they stop the synthesis of all prostaglandins, and many prostaglandins are important for maintaining the inflammatory process, such as prostaglandin I2, rather than just causing the pain. Okay, so non-steroidal anti-inflammatory drugs, which for short are just abbreviated down to NSAIDs. Okay, like so. Okay, right. So let me give you some examples of non-steroidal anti-inflammatory drugs. Okay, so the famous examples then of non-steroidal anti-inflammatory drugs, probably the most famous example is aspirin. Okay, another very famous example is ibuprofen. Okay, and some other commonly used drugs are indomethacin, okay, and also diclofenac. Okay, now all of these drugs work by stopping the production of the prostaglandins. Now, how do they stop the production of the prostaglandins? Well, by inhibiting the cyclooxygenase 2 enzyme that is induced in cells in these inflamed areas. Okay, so if you inhibit this enzyme, you can't convert the arachidonic acid into prostaglandin H2, and the prostaglandin H2 is the thing out of which the other prostaglandins, so prostaglandin E2 and prostaglandin I2, are produced. Okay, so you block the production of all prostaglandins, and that's a key element of the inflammatory milieu now lost, basically. Okay, the loss of things like prostacyclin is going to lead to the anti-inflammatory effects of these drugs, and the loss of prostaglandin E2 is then going to lead to the analgesic effects of these drugs, the ability to relieve pain. Okay, I should have just defined that at some point. An analgesic drug is a drug with the ability to relieve pain, okay? And we'll come back to this later on when we come to local anaesthetics. An anaesthetic is also a drug that has the ability to relieve pain, okay? But we'll talk about the difference potentially between those two a bit more later on. Okay, right. Those are the major non-steroidal anti-inflammatory drugs, and they work by blocking cyclooxygenase 2. Now, there is one thing that I need to mention here, which is there is a major drug that is classified as a non-steroidal anti-inflammatory drug, which is not in that list, but is hugely used. And this is the drug paracetamol, okay? Which is also called acetaminophen in the States. So paracetamol is equal to acetaminophen. Okay, now this does not work by blocking cyclooxygenase 2. Its ability to block cyclooxygenase 2 is really quite minimal. Okay, it also does not have anti-inflammatory effects like all the rest. Okay, so it must work in some different way other than blocking the production of all prostaglandins. Otherwise, how would you explain it not having the potent anti-inflammatory effect that all the others have because of the blockage of prostaglandin production such as prostacyclin? Okay, however, it does have a very powerful analgesic effect. Okay, and I say very powerful in quotation marks. It's not particularly powerful compared to the opiates, which we'll come on to later, but it has a considerable analgesic effect. Okay, now the mechanism of paracetamol, unfortunately, is not really known. Okay, some people suggest that it works by inhibiting COX-3, which is a third form of cyclooxygenase, which isn't really a separate gene product, but in fact is just another splice variant of the gene for COX-1. Okay, so it's a splice variant from the COX-1 gene, basically. So the COX-1 gene can produce both COX-1 and COX-3. Now, COX-3 is found in the brain, and it's suggested that maybe paracetamol or acetaminophen's effect is through blocking that enzyme in the brain. Okay, but really, the mechanism of paracetamol isn't understood at all, really. Okay, so the point of that was saying not this one, basically. All of the ones on top work in this way, but this one, which is counted as a non-steroidal anti-inflammatory drug and is hugely used, okay, one of the most used, we don't actually know how it works, really. Okay, right, so that's the non-steroidal anti-inflammatory drugs now complete. What I want to now move on to is the effects of another molecule that is within the inflammatory milieu, okay, and this is a molecule known as bradykinin, a very important peptide molecule present in inflamed tissues at high levels. Okay, a very important part of the inflammatory milieu. And like the prostaglandins, or specifically like prostaglandin E2, it is also capable of both activating C fibers directly, okay, or nociceptors attached to C fibers, the polymodal nociceptors, okay, and therefore causing them to fire action potentials, and also it's involved in the sensitization leading to allodynia and hyperalgesia, and again it does that in all the different nociceptor types. Okay, so we want to have a look at these mechanisms. Now, the first thing I'm going to say is that we don't know how it actually causes the direct activation of the polymodal nociceptors. Okay, so we don't know how it causes activation of the nociceptors that leads to firing. Okay, that mechanism is poorly understood. But we do have an idea about how it can cause sensitization. Specifically, it also leads to thermal allodynia and thermal hyperalgesia by activating the TRPV1 to open at lower temperatures, basically lowering the threshold potential for TRPV1. So let's have a look firstly at the synthesis of bradykinin and where it actually comes from in the inflammatory milieu, and then what we'll look at is how bradykinin actually works on neurons, and we'll have another G protein signaling cascade. Okay, right. So bradykinin then, so it is synthesized from a precursor known as kininogen. Okay, so kininogen is the precursor. Now, kininogen is found within the blood, okay, and it's put into the blood by the liver. So the liver is all the time synthesizing this precursor kininogen, and the kininogen usually circulates in the blood and can't leave the blood. Okay, but of course, when you've got an inflamed area of tissue, okay, and I'll draw my little box again to represent my inflamed area of tissue, you get the inflammatory process occurring, and in this inflammatory process you get endothelial cell contraction within the capillaries and also the postcapillary venules, and this allows proteins to leave the lumen of the bloodstream and go into the tissue fluid. So kininogen now ends up in the tissue fluid. Okay? Right, so what is actually now going to convert it into bradykinin? Well, this unfortunately doesn't have a simple answer, okay? It's one of these awful cascades involving loads of serine proteases activating each other, okay? It's not quite as bad as the coagulation cascades, okay, but it is still called a cascade, and in fact it's called the Kine cascade, what I'm about to tell you, okay? But as I say, it's not as bad as the coagulation cascades, okay, as far as learning about it is concerned. So, right, so what happens then is the first enzyme that is going to be activated in order to set off this conversion of kininogen into bradykinin is what's known as factor 12, which is the same factor that's involved in the coagulation cycles. Coagulation cascades rather. Okay, so factor 12 here, okay, which also has another name that some people will call it. It's also called Hagelin Factor. Okay, and basically Hagelin Factor, again, it's a protein that is produced by the liver. Okay, in fact all of these proteins are going to be produced by the liver initially, and it usually circulates in the blood perfectly happy, not doing anything. Okay, and then when it gets to a site of inflammation, it can suddenly come out of the blood and go into the extracellular fluid, and then it meets molecules that it would absolutely never, ever, ever, ever be allowed to meet if it was in the bloodstream. Okay, and one of these molecules is collagen. Okay, so collagen is a key activator of factor 12. So collagen is everywhere in the extracellular fluid, okay? It's a key component of the extracellular matrix. So you have collagen fibers all over the place. So it's not going to take too long for the Hagelin factor to meet a collagen fiber, basically. Okay, and when Hagelin factor meets collagen, collagen leads to the activation of the Hagelin factor. So it now becomes factor 12A, the active factor 12. Okay, or you can call that activated Hagelin factor depending on your taste. Okay, so we now have this active enzyme which is factor 12A, which has been activated by bashing into a collagen molecule. Okay, right. Now what can happen is this enzyme can now activate another enzyme, and the enzyme it's going to activate is prekallikrein. So again, prekallikrein is produced by the liver, it's circulating in the blood, doing nothing, okay, but now at this site of inflammation it can leave the blood and go into the extracellular fluid, and now factor 12A is activated, and it can now convert prekallikrein into the activated form. Okay, so prekallikrein can be converted into the active enzyme, which is just called kallikrein. Okay, right, now the kininogen that has come into the extracellular fluid from the blood is going to be converted into bradykinin by kallikrein. Okay, so it's not too long a cascade. So kallikrein is now going to convert kininogen, okay, into bradykinin. Okay, so that's how you get the production of this little peptide, bradykinin, at sites of inflammation, okay, by this cascade here. Okay, so now we've got the production of bradykinin. So let's now see what effects bradykinin is actually going to have on these neurons. Okay, and I'm afraid this involves another one of these G protein cascades. Okay, this time the GQ cascade. Okay, right. So we'll go over the page. Right, so neurons have on their surfaces then receptors for bradykinin. Okay, and I'll draw this like so. Here once again is the plasma membrane represented as these two parallel lines. Okay, and again we'll have a G protein coupled receptor, which is a receptor with this characteristic 7 membrane spanning alpha helix structure. And this G protein coupled receptor is going to be specific for bradykinin, and the one which nociceptors express is known as the B2 receptor for the bradykinin type 2 receptor. Okay, so here we have our bradykinin type 2 receptor. Now, when bradykinin binds to this, it's going to trigger a cascade which we'll go through in a moment. Okay, but once again I want to draw your attention to the point that bradykinin is capable of doing two things. It's capable of actually activating polymodal nociceptors to fire action potentials, and also it's capable of sensitizing all nociceptors, okay, to produce the allodynia and hyperalgesia. We do not know how it actually causes the firing of action potentials. That bit remains unknown. Okay, what we do know is how it causes the sensitization, so we're only going to see the bit regarding the sensitization, and specifically all we know at present really is the sensitization of TRPV1, okay, leading to thermal sensitization, thermal allodynia and thermal hyperalgesia. Okay, right, so again when bradykinin, which I'll just abbreviate down to BK, binds to this B2 receptor, the B2 receptor is going to change conformation, and it's now going to be able to activate a heterotrimeric G protein. Okay, now the heterotrimeric G proteins that B2 receptors are capable of activating have as their alpha subunit a subunit known as the alpha Q subunit here. Okay, so then also alongside it, of course, we have the beta-gamma complex, and again we don't care which beta subunit and which gamma subunit you have there. Okay, now initially it's in the off state with GDP bound, but now that the B2 receptor has been activated, it is of course going to bind to this heterotrimeric G protein and activate it. Okay, so we'll colour this in. Here's the alpha Q subunit in red, here's the beta subunit in blue, okay, and here's the gamma subunit. So now what's going to happen is the B2 receptor that's activated is going to bind to this GQ heterotrimeric G protein, and remember, heterotrimeric G proteins are named after what the alpha subunit is, so this is a GQ heterotrimeric G protein. So it'll bind to GQ heterotrimeric G proteins. It will then catalyze the conversion of the alpha Q subunit from being in the off state to being in the on state. So it will catalyze the release of this guanosine diphosphate molecule and the entry of a guanosine triphosphate molecule. So GDP will leave and GTP is going to come in. Okay, that will then cause the change in conformation in the alpha Q subunit that causes it to go from being in the off state to being in the on state, and then when it's in the on state it's going to cleave away from the beta gamma complexes. Okay, so now you'll have your alpha Q subunit with GTP bound in the on state, no longer with the beta gamma complex here. Okay, so here is the beta gamma complex on its own here. Okay, so let's colour in these subunits once again. So here is the alpha Q GTP complex, and here is our beta gamma complex over here. So now, what does the alpha Q complex do? Okay, it is going to activate enzymes known as phospholipase C beta enzymes. Okay, so let me write this here. So phospholipase C beta enzymes. Okay, so there are absolutely huge number of different phospholipase C enzymes. One family of phospholipase C enzymes is the phospholipase C beta enzymes, abbreviated down to PLC beta. Okay, and all of these beta family of phospholipase C enzymes are those that can be activated by the alpha Q subunit when it's in the on state. Okay, so other families of phospholipase C enzymes, such as phospholipase C gamma family, they are not activated by the alpha Q subunit. Okay, they are activated by receptor tyrosine kinases. Okay, right. So basically, phospholipase C beta enzymes, they are continuously moving between being in the cytoplasm and being attached to the underside of the phospholipid bilayer, so they can bind to components of the phospholipid bilayer. However, this is just moving around basically. They're not actually gaining activity by doing this. They're always inactive, okay? So these two here that I'm drawing, they're inactive here, but they can just move around. They can either be floating in the cytoplasm or they can just temporarily be bound to the membrane, but they're not doing anything. They're just bound to the membrane in a different location. The alpha Q GTP subunits can bind to the phospholipase C beta enzyme when it is bound to the membrane in this way, and can stabilize it being bound to the membrane, so it can hold it there, and can also activate it so that it now gains its catalytic activity. Now, what is the catalytic activity of a phospholipase C enzyme? Well, this is the same for all phospholipase C enzymes, whether it's beta family or not, okay? And what they do is they break down the membrane phospholipid phosphatidiol inositol 4,5-bisphosphate into IP3 and diacylglycerol. Okay, so let me write this down. So, PIP2 stands for phosphatidiol, that's the P, okay, then the I is for inositol, okay, and then it's 4,5, and then the P2 means phosphate twice, so 4,5-bisphosphate. Oh dear, running out of space here. Right, so phosphatidiol inositol 4,5-bisphosphate, which more properly should be abbreviated really down to PI45P2. Okay, right, so let me just talk you through the structure of this molecule. It is a membrane phospholipid. It's within the phospholipid bilayer. Okay, right. So basically, let's start off with the phosphatidiol prefix. Okay, what that means is that the molecule is based on the structure of another molecule known as phosphatidic acid. Okay, now let me firstly tell you about what the structure of phosphatidic acid is. Now I'll draw this within the phospholipid bilayer. Okay, so here's the phospholipid bilayer zoomed in on. Okay, and now I'm going to draw your phosphatidic acid molecule in cartoon form, of course. Okay, right, so let me highlight up different components here. So these two vertical lines coloured in orange here, these represent long-chain carboxylic acids, or fatty acids as a biochemist would call them. Okay, these are just carboxylic acid molecules with really long hydrophobic tails, and these are the lipid component that is associating with the other lipid components of the phospholipid bilayer. Okay, then this horizontal line, now coloured in green here, this represents the glycerol molecule. Okay, now glycerol is a free carbon molecule with alcohol groups coming off each of the free carbons, well, one alcohol group coming off each carbon. Okay, and basically onto the first carbon's alcohol group and onto the second carbon's alcohol group, you have attached the long-chain carboxylic acids by ester links. Okay, then onto the alcohol group that comes off the third carbon, you have attached a phosphate group, which is this purple blob here, okay, via a phosphoester link. Okay, so this is why phosphatidic acid is considered a phospholipid. It has the two components that you need in order to be a phospholipid, which is a phosphate group and a lipid group, and you've got two lipid groups, so it certainly fulfills that criterion. Okay, right, so that's phosphatidic acid. How do we convert this into phosphatidyl inositol 4,5-bisphosphate? Well, basically what we do is we add on an inositol ring, and the way I cartoonly show an inositol ring is just a hexagon like so. So this is inositol. Okay, and inositol is a 6-membered carbon ring where each of the carbons has an alcohol group coming off it and also a hydrogen coming off it. So all of the carbons are singly bound to one another, okay, and every single carbon in this ring also has an alcohol group coming off it and a hydrogen coming off it. So the carbon here, its alcohol group is bound to this phosphate group via a phosphoester link. Okay, so we'll colour the inositol ring in blue here. So that's phosphatidyl inositol now. To convert phosphatidyl inositol into phosphatidyl inositol 4,5-bisphosphate, we then just stick a phosphate group coming off the alcohol group of the 4th carbon here, and then a phosphate group coming off the alcohol group of the 5th carbon here. So we've counted round 1, 2, 3, 4, 5. Okay, so that's in phosphatidyl inositol 4,5-bisphosphate, or PIP2, cartoonly shown here. Okay, so it is a phospholipid. It's a normal component of the plasma membrane. It's not a particularly common phospholipid. It's quite a rare phospholipid in the phospholipid bilayer. Okay, but it is present. Okay, now what's going to happen is our phospholipase C-beta enzyme, now that it's active, is going to break down this phospholipid into 2 separate molecules. Okay, now what it's going to do specifically is it's going to cut this bond here. Okay, between that phosphate group that comes off the 3rd carbon of the glycerol molecule and the 3rd carbon of the glycerol molecule. Okay, that's where it's going to cut, and it's therefore going to produce 2 products here. So let me draw out the 2 products. One of them is going to be the glycerol molecule with 2 long-chain carboxylic acids bound to it, like so. And this molecule is known as a diacyl glycerol molecule because it's a glycerol molecule with 2 acyl groups attached to it, which is just a name for the portion of a carboxylic acid molecule that you attach onto an alcohol group when an ester link is formed. Okay, and for short, diacyl glycerol can be abbreviated down to DAG. And then the other molecule that you produce is inositol, shown here, with the 3 phosphate groups coming off it, off the 1st, the 4th, and the 5th carbons of that inositol ring. Okay, so we'll colour this in. Here is the inositol ring in blue. Okay, and here are the 3 phosphate groups coming off it. Okay, and that molecule now is called inositol 1,4,5-trisphosphate. So inositol 1,4,5-trisphosphate. And for short, inositol 1,4,5-trisphosphate is usually abbreviated down to I for inositol, P for phosphate, and then because you've got 3 of them, IP3. Okay, so that's inositol 1,4,5-trisphosphate. Okay, right, so what do these two then do? Well, let's start with IP3. IP3 leads to the release of calcium from intracellular stores by activating IP3 receptors. Okay, so I'll just add that on just to complete this down. But actually, the one that's going to be important to us in this case is actually the diacylglycerol. Okay, so I'll just draw our IP3 receptor here, which is another ion channel present in the membrane of this intracellular organelle, the endoplasmic reticulum here. Okay, so this is our IP3 receptor, which for short I'll just abbreviate as IP3R, and IP3 binds to the IP3R and leads to the opening of the IP3 receptor. The gating mechanism of the IP3 receptor is still up for debate, okay, but to keep this basic, IP3 leads to the opening of the IP3 receptor, and therefore calcium is released from the endoplasmic reticulum. Okay, right, since the IP3 receptor is a calcium channel and the ER contains a large concentration of calcium whilst the cytoplasm contains very, very little calcium. Okay, so that's the two key parts of this signalling pathway. You get a calcium signal and you get diacylglycerol produced. Okay, now these two are going to be able to activate protein kinases known as protein kinase C enzymes. Okay, and these protein kinase C enzymes are going to be able to phosphorylate trip channels just like protein kinase A could, and again sensitize it so that it responds, i.e. opens at lower temperatures than normal. Okay, now most protein kinases actually, well, the main protein kinases require calcium and diacylglycerol to activate them, okay, but the one that we're actually going to see is involved in phosphorylating TRPV1 channels actually doesn't require calcium. Okay, so let's just have a little bit of a discussion of the protein kinase C enzymes then. So, protein kinase C enzymes. Okay, so there are three major families of protein kinase C enzymes. Okay, there is what's known as the conventional protein kinase C enzymes, which are abbreviated as the CPKCs. Okay, and this stands for conventional here. Okay, and these are the main ones. Okay, so these include three different enzymes: protein kinase C alpha, protein kinase C beta, and then protein kinase C gamma. Okay, so those are the three types of protein kinase C enzymes, which are the conventional protein kinases. Okay, the next group of protein kinases is what's known as the NPKCs, which are the novel protein kinases. Okay, and these ones only require diacylglycerol and not the calcium to activate them, unlike the conventional protein kinases, which require both. Okay, so novel protein kinases include protein kinase C delta, and I'm just wondering, I'm not going to be able to actually fit in it written out in full like that, so I'll just put the Greek letters. Protein kinase C delta, protein kinase C epsilon, protein kinase C eta, protein kinase C theta, and also protein kinase C mu. Okay, so these ones are just activated by diacylglycerol. Okay, whereas these ones up here, the conventional ones, are activated by diacylglycerol and calcium. Okay, and then finally there is also the atypical protein kinase Cs, okay, which we're not going to see, but these include protein kinase C zeta and protein kinase C lambda, so the Greek letters get gradually more fancy. Okay, so these are the atypical protein kinases, and these just have random things activating them. So for instance, zeta is activated, if memory serves right, by ceramides. Okay, and I think lambda is activated by SRC tyrosine kinases. Okay, so the protein kinase C enzyme that's going to be important to us is protein kinase C epsilon, which is one of these novel protein kinases, not one of the conventional protein kinases. Okay, it's this one that we believe is responsible for phosphorylating TRPV1 channels. Okay, and this is going to be activated by diacylglycerol alone. Okay, so the diacylglycerol here is going to activate protein kinase C epsilon, okay, which I'll represent like so. So here's our protein kinase C epsilon, and this is then going to phosphorylate TRPV1 subunits in TRPV1 channels, leading to those TRPV1 channels now opening at much lower temperatures, and therefore leading to the thermal allodynia, basically. Okay, so that is what is understood of how bradykinin causes the sensitization of neurons, okay, specifically of these nociceptors. Okay, so this is going to work within the thermal nociceptors and the polymodal C fibers to sensitize them to thermal stimuli. Okay, there are most certainly other pathways by which bradykinin achieves this, but this is one of the major ones that is well understood. Okay, and as I'll say again, I'll stress the point that bradykinin is capable of actually directly activating the depolarization of polymodal nociceptors, although the pathway by which it does that is not understood at all. Okay, right, so in the next video what we'll move on to is discussing nerve growth factor, another component of the inflammatory milieu, and then we'll also discuss central sensitization before moving on to the local anesthetics.